Okay, I'm gonna go ahead and get started. My watch says 9:31. The wall on the clock. The wall on the clock. It's gonna be that kind of day. The clock on the wall is a little slow. I'm also gonna have to leave a little early because genomics candidate phone interviews all day. So today we're gonna start talking about single-cell RNA-seq, and then on Thursday we'll do metagenomics. Before I launch into the new stuff, any questions about the homework? I haven't looked at any of it yet. You should have been able to answer everything except number 5 with what was already in the notebook. So if I was really the only thing where that was sort of a next step of, okay, ask ChatGPT for something that you haven't been given yet. Curious how people went about trying to address that. What'd you do to answer that question? Ask ChatGPT, but how? How did you ask it? That's my question. Did you give it the Colab notebook? Yes? It doesn't really like Colab notebooks, though. Yeah, love it. It's so smart. It can read screenshot pictures. Other more direct ways, less computationally intense. You gave it the results and then asked it what the top 4 were?
Yeah, I just asked what were the.
Oh, okay. Yeah, it probably can go online and find the answer for that one. That one won't work generally. It will work for the wine data set because that's a scikit-learn standard, been done probably millions of times. The issue, so getting the Colab notebook files into ChatGPT, if you want it to read what your code has done so far, the most straightforward way is to save it as just a .py file. So it gets all the code. And you can do that from Colab. When you go to download your notebook, it gives you an option for a .ipynb. That's the IPython notebook. That's the whole markdown format. That's what ChatGPT doesn't really like. It's much more straightforward and fast if you download it as a .py and then give it to ChatGPT. And .py is just the Python code. I don't think I've even looked at what it does with the markdown. But it doesn't matter. It doesn't have all the extra stuff in there. So then how many of you got code from ChatGPT, put it in the notebook, and then had it give you the table and you pulled out the top? Yeah, okay. That's good. Most people nodding their heads. So ideally, that's the most broadly useful to you. Making it go retrieve the feature importance from the random forest, for example, and then tell you, because that's a common question you might want to have. What aspects of my data give me the most information about the thing I'm trying to predict? Okay, so unless there are other questions about that homework, I'm going to move to single cell. So the new module has only a PowerPoint today because you have to make your own Colab notebook. This is the next step in becoming like learning to learn to do bioinformatics rather than just learning bioinformatics. So if we get that PowerPoint, it's not that one. That's last week. It is this one. Right, so last week, we trained models to classify samples when we already knew what the labels were. So in the wine data, anybody look at what the labels were? I mean, it's in some of the figures, but actually, anybody remember what your classifier data was? It's been a whole weekend. Actually, it was just last night that the homework was due. What were you trying to predict? Those were the features, but what were the things that, like, what was the label? Yeah, the type of grape, cultivar, whatever you want to call it. And how was that labeled? There wasn't anything in the notebook that actually had you do it, right? It did like x dot head. X are the features. Y are typically the labels. And so it used the labels to in some of the figures, but it's not super intuitive because the labels are not at all intuitive. If you're expecting grape names, you won't find them. It's just 0, 1, 2. It's like class 0, class 1, class 2. There's nothing interesting about the labels, and I think that maybe that was intentional or not. I don't know. So in any case, your data were labeled last week. So when you did the clustering, you were trying to find the model that basically partitioned those clusters, those known breaks in your data the best. This week, we don't know what the labels are. So we're going to use unsupervised learning approaches to find whatever the structure is in the data, assuming there's some structure. And then we'll go back and use those cluster labels to try to predict. In this case, we're going to use the same data, but you could use it to see whether your clusters can predict new data. Basically going back and completing the cycle that we did last week. All right, so how many of you know anything about single cell genomics, transcriptomics, and ever dealt with this data? No? Read papers about this kind of data? This is the way, at least the genomics world that I live in, this is the way everything is going. You essentially start out with some tissue. This arrow is a ton of work in figuring out how to get the individual cells dissociated from each other. In case I didn't really appreciate going into our first studies of this, how sticky cells are. They just like to stick together. The reason it's important that you get them dissociated is that the next step is you put them on this microfluidic device, and this is like a super zoomed in picture of the device, where you can imagine each of those colored dots in the picture is a different cell. And so the width of that channel is roughly the width of a single cell. And you're feeding in chemistry, basically, from these other channels that go perpendicular to your line of cells. So you're forcing cells through this microfluidic channel. The enzymes or whatever chemistry you want to add to it is also flowing up into that mix. And then the next channel is something more viscous. It's usually some kind of oil. And what that does is form little bubbles that ideally have one cell and one sort of bead of chemistry. Okay, if you zoomed way in on what's going, actually, I said that wrong, didn't I apologize. Cells are going, whoops, the cells are in this channel, and it's the beads that are going across there. So ultimately what you get at the end is what I was about to say, but I realized the dimensions of those circles are wrong. Right, that little bitty thing is the cell, and then you've got this bead. If you zoom in on it, it has all of these little furry things are little oligonucleotides that are going to grab onto messenger RNAs. Okay? In the case of RNA-Seq, okay, you can do this for genomic as well. Those little oligos, much like we talked about when with multiplexing and regular RNA-Seq and stuff like that, they just have a barcode on them. And they say something about which cell did it come from, okay, and which molecule is it reading. So what you end up with is everything that has the same set of barcodes, for example, you know all of these messenger RNAs came from the same cell. This, I should also say, this is a generic view of how single cell works. There are multiple companies out there. 10x Genomics is probably the one that's being used most commonly by the faculty here. But especially if you work on model systems where you have known marker genes. So you can do flow sorting where you have a label inside the cells that you want to retrieve, okay? And so that is a way to sort of enrich your cell population for certain kinds of cells. This workflow that I'm describing is going to get all the cells in whatever tissue you give it. So depending on how heterogeneous the cell types are in that tissue, that's going to determine a lot about how many different kinds of clusters do you expect to get at the end, right? Okay, so anyway, all those things get tethered to the bead. There's some chemistry that goes on. This is why I went into the tangent about the different methods, is that this arrow is also a lot of work, but that work is usually outsourced to a company or a core facility that has the machine to do their chemistry. Okay, so going from this little bead emulsion, and in the case of 10x, even that, usually you're just sending them the cells, and you can either get back the library, right, the Illumina library usually, and then sequence it here at the North Texas Genome Center, or lots of companies, you just send them the cells and you get back the sequence. All of the demultiplexing that tells you how many cells you had, how many reads per cell, all of that stuff usually happens at the core facility that does the sequencing. So we're sort of skipping over these steps that are, these are the most stable in terms of like the pipeline. They just push a button and it runs, and it gives you back your FASTQ files that have the sequences for each cell. For 10x, the pipeline is called Cell Ranger, or the software package is called Cell Ranger, if you come across that in the literature. And then, so what we're really going to focus on is what happens after you've got that data, and how do you identify the cell types based on that, and differential expression. Just a quick word about this field is moving so fast, it's super hard to keep up with. Like, this is the frontier, and I feel like everybody knows this is what you need to be doing. and there are all kinds of problems with it still. So that's the times when science kind of moves the fastest, because everybody's trying to solve all the problems. One of the areas that is the most recent are the spatial multi-omics methods. So this is where, right, when you've done whole tissue homogenization to get your cells, you don't know where those cells came from in the tissue. Okay, what we'll see later is we can sort of predict what the cell types were, so you can sort of backtrack if you know something about the organization of the tissue, but you don't have that information going into the experiment. There are now methods that are essentially these plates that have nanowells in them. You take a super thin slice of your tissue, put it on the plate, and suck the cells down into that plate. So now your little bubbles of chemistry know spatially where they were relative to each other. Okay, and then you can do all the reason I don't put transcriptomics only is that there's more than just RNA-seq you can do with this. You can do ATAC-seq, which is treating it with an enzyme to look at where the chromatin is open or not. So you can look at regions of the genome that are being expressed. Even if I said it now, by the end of the day, there's probably a new one. Way to do multi-omics. So just be aware, and then like the laundry list of different packages and softwares that get used to analyze this stuff, right? Also, it's growing as fast as the wet lab methods. Probably faster than the wet lab methods.
When you do your single cell stuff, what is, I guess, the next thing that you usually pair it with, like in your lab? Or do you usually just do the single cell RNA-seq?
I'll show you in a second. So this is the generic, like if you're interested in human, one of the early single cell studies. And when I say early, this paper is 2019. So this is what being on the frontier is. And we're going to look at data that is like this. So this is from blood samples, I believe, from arthritis patients, trying to look at variation in immune cell gene expression. And so what this study showed, first pass clustering, right? You get these fibroblast, plasmablast, B cells, T cells, as sort of major breaks in terms of classification of cell types. But then even within B cells and T cells, for example, this was one of the main points of the paper, you see variation in subpopulations in their gene expression. And so then you can compare, say, the distribution of subtypes of T cells between an arthritic patient and a non-arthritic patient to potentially come up with some kind of treatment if you can understand why those genes are being expressed differently in those subtype populations. This came straight out of the paper, but what we will get to by the end of today is you being able to make all these plots. Now, well, this one probably made in PowerPoint or like making the ellipses with the labels on them. The clustering though, and then you could do subclustering. This is showing, for example, the gene THY1. This is showing within the fibroblast cells which genes were expressing. So the darker green means higher expression. Every dot there is an individual cell. And so you're getting the readout for, you know, if you look across all the different cells, and like HLA-DRA is expressed in all of this type of B cell, but not, or a very low level in this type. Okay. Stuff like that is what you can get out of there. For my lab, back to Adam's question, this is one of the recent papers out of my lab looking at spermatogenesis in the beetle that we mostly work on. And we went into this experiment trying to figure out what happens to X chromosome expression. You can imagine maybe. When you go through meiosis and you've got a chromosome that doesn't pair with anything else, that might be a problem if it's still active. And so, anyway, we're interested in what happens to gene expression for the X chromosome. But nobody knows anything about what's going on in beetle testes. So we did the single cell experiment. This is that first pass clustering. There's a lot that goes into this. But then you don't know the right answer here, right? We don't know how many cell types should we be able to recover. We don't know how synchronous spermatogenesis is in beetles. I mean, we have some idea from microscopy studies, but in terms of gene expression, no clue. So this you sort of treat as a hypothesis. This is the most stable clustering that we could get. We'll talk about the parameters and all that go into that in a second. Given that clustering, does it make any sense? And so we did things like looking at what cell phase they're in, as they go through mitosis and meiosis. We did ploidy, and this is the one that said, actually, we probably do have some meaningful clusters here because our haploid cells, the ones that have finished meiosis, all sort of clustered together in things that we were calling probably post-meiotic cells. So that was pretty comforting. And then you sort of backtrack once now you think, okay, my cells do have some biological meaning. Then you look at what are the pathways that are regulating spermatogenesis in the beetles. Does that answer your question, Adam? What would I do with that?
No, I think my question was like, so like, because you're talking about multi-omics, right? And so when I think of single cell RNA-Seq, like I think about the process that you talked about where you like isolate like the individual cell, get the RNA per cell, but then there's also like the spatial transcriptomics that you talk about, like where you like get like a broad overview, and I'm wondering if those two are paired together. And so I'm wondering, like, for this study, did you just do the single cell RNA-Seq, or yeah?
We did, and whether you're pairing them together after the fact or doing them at the same time, I think is most people today, if they're gonna do both kinds, are doing them at the same time. So you've got the ATAC-seq, the RNA-seq, and the spatial element all from exactly the same cells. Yeah, it would be possible. Like if I went, I got some money to do a spatial RNA-seq, I could map the clusters from this experiment onto those, maybe boost my gene expression power to detect differences, but yeah. Just to make some connections between what we did last week and this week, I already talked about how we had training data with known labels. This week we're going to discover unknown cell types. The visualization, PCA and UMAP, are essentially the same. And then the model evaluation metrics are a little different, right? We had this cross-validation and the train and test data to sort of get estimates for the accuracy of our models. In this unsupervised approach, where we're just trying to discover whether we have good clusters, we're going to use marker genes to assess whether these things are meaningful or not. Okay. And that's sort of analogous, in no way mathematically analogous, to marker genes being the labels, and the confusion matrix being some metric of validation relative to there's a high level of this marker gene in this cluster, and therefore it has biological meaning. Yeah. So what we're actually doing, we only had 13 traits for our wine classifier. We've got thousands of gene expression levels for the RNA-seq data. Just much bigger data. And potentially more clusters, but we don't really know how many clusters to expect, right? No labels. And last, just in terms of when we had labeled data and we were looking for the accuracy of our models, we had this grid search algorithm where we tested multiple hyperparameters to see which ones gave us the best predictors, okay, or the best model that gave us the most accurate predictions. When we go to scanpy and we're doing the cluster identification, there are a whole sort of different set of hyperparameters. And so you test multiple hyperparameters to, again, I mentioned this a minute ago, to look and see whether the clusters are stable or not, okay? So by model selection here, we're choosing the settings that produce stable clusters. And not just stable, meaning the graph isn't changing around, but also that the marker genes are expressed in the right cluster and not expressed in other clusters. So I've already been using so much jargon. Marker gene is a huge one. I didn't put a vocab slide in this one. You guys what's a marker gene? Just from the context. Just make sure you're tracking with me here. What do you think a marker gene is? Mostly right, I think. Yes, it's a gene that has a known expression in a certain kind of cell, right? It's a marker for that cell. And so when we do our clustering, then we'll say, okay, show me where this gene is expressed. We expect to see it only expressed in one cluster. That tells us our clustering is good because it does a good job clustering something that we have a marker for, okay? So that correlation between a marker gene and a cluster is what's telling us whether we have good clustering or not. But like the flip side of that is I've done clustering because the algorithm is going to cluster based on expression profiles. But if you have a known marker gene that should only be expressed in one cell type, but you see that it's expressed in all different clusters, your clustering algorithm is not doing a good job, okay? All right. The kind of clustering that you almost always see in single cell analysis is called Leiden clustering. I've got it in terms of words there. The most important thing I put in red, lower resolution, fewer larger clusters, higher resolution, more smaller clusters. I think it's easier to think about this in terms of the graphic. What this is doing is it first builds this graph, right, where where it's going to try to cluster the cells that have the shortest edges. The edges are indicative of how similar is the gene expression overall, okay? So what it does is start to break up that graph into modules or community. They're often called communities in Leiden clustering. I don't know where that terminology came from. And so what tuning the resolution does is if you have a low resolution, right, that means you're going to have a few large clusters. It's going to split this less. And as you increase the resolution, you will get more clusters. Okay, and so finding the right resolution that is meaningful is sort of part of the challenge here. And how am I going to assess that? Like, what's meaningful? Like, what's a good resolution? How do I judge? Yeah, that's so. This really is a kind of a tricky part, but we just got done talking about marker genes, right? And so, if, for example, I have a known marker gene and I say, show me, let me go back to, right, that's one of the things these plots are really good for, okay, is this is showing individual genes overlaid onto the clustering, okay? And so if I went here and my marker gene only lit up, right, this bottom section of cluster 0, I might say, well, actually, I should probably use a higher resolution because it's not breaking this giant cluster up enough, okay? And so this clustering scheme, now that's highlighting what it's calling cluster 0 here. And so maybe this is a good one, okay? But then if I say, well, maybe I could turn up the resolution even more and recognize more cell types. Well, now when I highlight that, it's going to give me marker gene expression in cluster 0, cluster 4, cluster 11. That's no good either. So I've gone too far. Okay. Yeah.
Two questions. One, what do you mean by resolution? Like, is it like the sequencing depth? Is it?
It's how many breaks is it gonna? Well, what it's actually doing is saying what's the threshold for the length of the edge before I break it into a new cluster.
Okay, so that's like just a bioinformatics.
Yes. It's purely tuning the latent clustering. So if you have a high resolution, it breaks it on shorter edges. Okay, so that you'll get way more breaks in the graph. Yeah.
How do you identify the marker genes? Do you have to do like a
You have to work on a model system. Yeah, no, and so, back to my lab's thing, we didn't have good marker genes. We tried to use the Drosophila spermatogenesis marker genes, and there is no clustering parameter set that you can do that gives you good clusters based on the Drosophila marker genes. So we published that paper, and then we used that to get money to now go back and do the microscopy and the things that you have to do to say the wet lab part of our clusters actually are meaningful. Because now, you can treat this at the end of the day as a hypothesis. So I can tell you, based on the bioinformatics, what the marker genes are now for all these cell types in Tribolium. Like we now have the Tribolium spermatogenesis atlas, but we don't know if it means anything in a beetle testes. So that's what the microscopy you have to do. So you make an RNA probe, and you slice the testes and you label it and put it under a high power microscope and see, is it lighting up specific kinds of cells? So that's the validation, the real end of the day validation.
And the probes are based on the expression data.
Yeah, so I would say like this, whatever that is, teal early spermatid 2 cluster here, I could say what's the gene in that cluster that is expressed only in that cluster. And then I make an RNA probe that matches it, and I hybridize that to my tissue sample and look at it under the scope. And hopefully it's not lighting up the whole testes. It's lighting up one little subset of cells that look like early spermatids. Okay.
On the microscopy side, are you using a fluorescence or
Yep. That is like this is where science is like a collaborative process, because my lab doesn't do microscopy really. This is the Coromilla lab helping us out with this one. So yeah, we gave her the sequences of the marker genes and said, do your thing. And we'll give you the testes too, but let's see. I think that's laden clustering. Okay, so this is what we're going to do the rest of the day. Start your own CoLab notebook, and you're going to work through the scanpy basics. I'm going to pick up here on Thursday, but if you are able to finish that between now and then and you want to get a little bit of a jump start on the homework, you can look at the last slide and see if you can figure out how to add that code. Basically doing what we did last Thursday, right? At the end of the working through basics, you're going to have clusters for the data that should be biologically meaningful. This is human, I think it's blood. And then the question will be, well, do those clusters actually allow us to predict the new data? If we gave new gene expression data from single cells, could it predict what kind of cell they came from? Okay. All right. So I'm turning you loose to work on this. You'll probably hit a snag right out of the gate. And that is setting up the CoLab environment for this one. But the way to do this, this website is set up as a CoLab notebook effectively, but you can just copy that, paste it into your CoLab notebook, and try to run it, except that cell is not going to run because it doesn't have the packages installed. So you're going to have a pip install cell before that.